This portion of the basement membrane is made up of, again, the composition is same as that of the lamina rara externa, and the adhesion proteins are also present here, that is the laminin and fibronectin. But these adhesion proteins are responsible for the adhesions of these endothelial cells to the basement membrane. So these are the anchoring protein here for the endothelial cells to the basement membrane. Now in this picture, another important thing is present here, and these are the mesangial cells. These are the special type of cells, and this is the mesangial matrix, which is secreted by these mesangial cells, and collectively these are called the mesangium, and these are enclosed, you can see, on both sides by the basement membrane, and their function we will discuss shortly later. Okay. Now, just to revise what I have told you in the morning session about the glomerular basement membrane, that this is a common basal lamina for the podocytes and capillary endothelium. Thickness is about 300 nanometer in adults and 150 nanometer in children. And this glomerular basement membrane, it consists of three layers: the lamina rara externa. This is the electron-lucent layer adjacent to the podocyte process, and mainly composed of the glycosaminoglycan heparin, and which is negatively charged, so repel negatively charged organic molecules back to and block their passage through the GBM. Contain adhesive glycoproteins, that is the laminin and fibronectin, which anchors the podocyte process to the GBM. The lamina densa, the electron-dense middle layer of the GBM, main component is collagen type 4, which form a network and act as a sieve-like filter and restrict the large molecules to pass through it. The lamina rara interna is an electron-lucent layer bordering on the capillary endothelium. Composition is same as that of the lamina rara externa. Adhesive glycoprotein anchors the endothelial cells to the GBM. Now coming to this picture or this slide, and this is the key slide for many functions of the renal corpuscles, but we are interested here in the intra and extraglomerular mesangial cells. You see here, these are the mesangial cells. These are the mesangial cells. Now, what are these mesangial cells? These mesangial cells, these are the special type of cells, and these are present within the renal corpuscle. These are not seen with under the light microscope, so we can see them only under the electron microscope. And the study under the electron microscope reveals that these cells, they contain, in addition to the routine cell organelles, the bundles of actin and myosin filaments, and these actin and myosin filaments are responsible for their contractility. So these are the mesangial cells, and these mesangial cells can be divided into the intraglomerular mesangial cells and the extraglomerular mesangial cell. So these are the intraglomerular mesangial cells, and this, in continuity, you can see these type of mesangial cells are also present here. Those are present outside the these glomeruli. So these are called the extraglomerular mesangial cells. Another thing, but you can see here that these mesangial cells, they cover these glomerular capillaries on that side, which is not covered by the podocytes, not covered by the podocyte here and in this way here. So they are covering that side which is not covered by the podocyte. Now, what type, how they function, what are the functions of these intraglomerular mesangial cells? They perform multiple functions. First function is that they perform the phagocytosis. Phagocytosis, what they phagocytose? They phagocytose macromolecules that get entry into this space. They phagocytose those macromolecules, and they also phagocytose the antigen-antibody complexes. Another function is to provide the structural support. As you see, these are present in the center of the renal corpuscles, and these cells are responsible for the maintenance of the shape of the renal corpuscle. So these cells are providing the structural support. Third function is the provision of the immune cells. That what type of immune cells? For example, IL-1, for example, the prostaglandins, for example, the platelet growth factors. So these immune cells, these are produced by these mesangial cells, and these immune cells are used when there is damage to this filtering structures. These immune cells are required here. Fourth function, they can regulate the blood flow through the glomerular capillaries. How they can control the flow through the glomerular capillaries? You know that they are present here. This is the glomerular. This is the basement membrane. They are present here, covered by the same glomerular membrane. So when these mesangial cells they contract, they squeeze this membrane, and they reduce the glomerular filtrate, and vice versa. When they dilate to become and reshape, they can increase the glomerular filtrate. So in this way, they control the flow of blood in the glomerular capillaries. They have also got the receptors for angiotensin 2 and the atrial natriuretic factors, and in response to these stimuli, they increase or decrease the glomerular filtrate. So they perform various functions, these mesangial, intraglomerular mesangial cells. Now, they are contiguous with these cells, and these are the extraglomerular mesangial cell. You can see histologically they are of same type, but these cells are called Lattice cells. Lattice cells. Their function is not well understood up till now, but these are the part of the juxtaglomerular apparatus. Now this juxtaglomerular apparatus, it has got the three components. The macula densa, other one are the granular cells, also called the juxtaglomerular cells, and the third component are these mesangial cells, these extraglomerular mesangial cells. So these all three collectively called the juxtaglomerular apparatus. We have discussed already about these extraglomerular mesangial cells. Now we will come to the discussion about the macula densa and the juxtaglomerular cells or the granular cells. First, we will talk about this macula densa. Macula densa, where it is present. It is present in the this distal tubule, the terminal part of the straight part, of the terminal portion of the straight part of the distal tubule. Now, what happens here? Here you can see there is a change in the shape of these cells. These are become columnar, and they are closely packed, giving a dense appearance, and so it is called the macula densa. So macula densa is present where? In the terminal part of the straight part of the distal tubule. What they are doing? These are the osmoreceptors. By osmoreceptors, we mean they can check the osmolarity of the fluid which is present in this space at this level in the distal tubule. For example, if there is decrease in the sodium ion, they can assess it, and then they respond in two ways. In two ways they respond. First, they reduce the resistance of the afferent arterioles. by secreting some mediators. They decrease the resistance of the afferent arterioles, and secondarily, what they do, they in a paracrine manner, they stimulate these cells containing the zymogen, in which the renin is present to secrete the renin. Now, what are these cells? These cells and these cells, these are, you see, what is this? This is the afferent arteriole, and you see the myocytes here. So, in the tunica media of these afferent arterioles, what happens there is change in the smooth muscles are changed. They become larger, and there are zymogen granules present containing what? Containing the enzyme renin. So renin is present in these things. Okay. Now, another thing you can see, the basal side of the macula densa here, they are in a close approximation with these juxtaglomerular cells, and in a paracrine manner, they stimulate these cells to secrete the renin. And now what happens here to these cells? Those are responsible for secreting the renin. Actually, these are the mechanoceptors. Mechanoceptors means they respond to pressure. So when blood pressure is reduced in this afferent arteriole, they are stimulated to produce the renin. Now, we have much heard about renin. What this renin will do? When renin comes to the bloodstream, what it causes? It causes the hydrolysis of the plasma protein angiotensinogen. A plasma protein, angiotensinogen, is hydrolyzed by the renin to the angiotensin 1. Angiotensin 1 then moves to the lung, where it converts into angiotensin 2 by the angiotensin converting enzyme that is released by the endothelium of the capillaries, of the lung capillaries. So once angiotensin 2 is formed, angiotensin 2, it is a potent vasoconstrictor, so it increases the blood pressure on one side. Then it also can stimulate the adrenal cortex to produce the aldosterone. And aldosterone in turn conserves the sodium and in turn the water from the distal convoluted tubule, and so again increases the blood pressure. And this angiotensin 2 has got some direct effect on the renal tubules also. So this is a very important and key picture to understand all these things. And now, what is this? Some idea, more idea about these mesangial cells. You see here, what is this? This is the capillary space, and these are the endothelium, and then this is the basement membrane. You see the basement membrane, and the mesangial cells are present here. And in this way, you know, when they contract, they squeeze the basement membrane and reduce the glomerular filtrate. Okay, now to talk about and to repeat, I have written all these things just to make remember all these things. A special variety of the cell present in renal corpuscles called mesangial cells and their extracellular matrix, collectively known as mesangium, and these are present in between the glomerular capillaries and cover those regions of the capillaries which are not covered by the podocyte process. I have already shown you in that figure. Mesangial cells cannot be identified in routine sections. Electron microscopic study shows the irregular shape cells like pericytes of the ordinary capillaries. They are enclosed by the GBM. Cytoplasm of the mesangial cells contain bundles of actin and myosin filament showing their ability to contract. And then the functions I have already told you that the phagocytosis, structural support, secretions of cytokine, and the regulation of the blood flow. And then the juxtaglomerular apparatus, I have already told you. You can see this is the location of this juxtaglomerular. This is the vascular pole, and at the vascular pole, there are the cells which are not shown here are the extraglomerular mesangial cell. Other component is the Macula Densa, which is here in the terminal part of the straight part of the distal tubule, and these are the juxtaglomerular cells in the wall of the afferent arteriole. Okay. Now, same things, it comprises of following three components: the Macula Densa, the juxtaglomerular cells, extraglomerular mesangial cells. These are also called the Lacus cells. This is what I have already told you, that this picture is also very important, because you can see the decreased blood pressure leads to the juxtaglomerular cells, mechanoreceptors, and they stimulate the juxtaglomerular cells for the secret of renin release. On the other hand, decreased blood pressure, decreased glomerular filtration rate, decreased sodium concentration, and the Macula Densa osmoreceptor, and they stimulate again the renin release. Once renin released, renin in the plasma, it causes the hydrolysis of the angiotensinogen to the angiotensin 1. Angiotensin 1 in the lungs converted to the angiotensin 2 by the angiotensin converting enzyme. Once angiotensin 2 is produced, this is the potent vasoconstrictor, and then it leads to the constriction of the peripheral blood vessels and can increase the blood pressure. And the other thing, this aldosterone release from the adrenal cortex, and it increase sodium uptake from the DCT, increase water uptake, increase blood pressure. And angiotensin 2 also have got some direct action on the renal tubular cells. This is again the Macula Densa, which explains that it is a specialized region of the distal tubule where the tubules come in contact with the afferent and efferent arterioles at the vascular pole of the renal corpuscle of its own nephron. In the terminal part of the straight part of the distal tubule, cells changes from cuboidal to tall columnar. Nuclei are closely packed and give the tubule darker appearance, hence the name Macula Densa. You can call it dense spot. Okay, the basal lamina of these cells is thin and discontinuous. Blunt process of these cells extend through the discontinuous basal lamina toward juxtaglomerular cell. The Macula Densa function as a sensor of the osmolarity of the fluid in the DCT. Decrease in sodium ion concentration in the tubular fluid, generally due to decrease in GFR, stimulate Macula Densa to produce chemical mediators which have two effects I have already told you. It decreases resistance in the afferent arteriole, which increases the glomerular hydrostatic pressure to increase the GFR toward normal. It also stimulates the juxtaglomerular cell to secrete renin in a paracrine manner we have already discussed. Now, the adjacent to the Macula Densa, the tunica media of the afferent arteriole contain modified smooth muscle cells called these JG cells. These cells are mechanoreceptors. These are large cells with spherical nucleus. Cytoplasm is full of zymogen granule containing this enzyme renin. And the internal elastic lamina is absent, so JG cell lie close to the endothelium. In response to decrease in blood pressure in afferent arteriole, the JG cells secrete renin, which is an acid protease, which causes the hydrolysis of the plasma protein angiotensinogen to angiotensin 1. Angiotensin 1 is converted into angiotensin 2 in the lungs by the action of the angiotensin converting enzyme, which is secreted by the endothelium of the lung capillaries. Angiotensin 2 is a potent vasoconstrictor, which causes an increase in the blood pressure. It also causes stimulation of the adrenal cortex to produce aldosterone to conserve sodium from the distal convoluted tubule. Now coming toward the last topic of today's discussion, and that is the filtration barrier. Now what you can see here, there are three things are here. These are the endothelium, and this is the, here you can see the, first you can see this is the blood site, where blood is present here, and the urine is present here. Okay, now the first thing is this, the endothelium, and it is covered by some glycocalyx. I will show you just later on. And then this is the basement membrane, which is the common membrane between the endothelial cells and the secondary process of the podocytes. In between the podocytes, you can see these slates. These are the filtration slates, and then these slates are covered by the membrane, and these are called the filtration membrane. Now, this is another picture, a more elaborated or more descriptive picture. Here you can see these are the endothelial cells, and this light color here, this is shown for the glycocalyx, and then the basement membrane. These are the secondary process of the podocytes. In between them, the filtration slit, and this is the 20 nanometer distance, and this is they are showing the slit diaphragm made up of various types of the proteins. Okay, now this barrier consists of three components. We already know the endothelium, the glomerular basement membrane, the slit diaphragm. What is the role of the endothelium? The glomerular capillaries are of fenestrated variety lined by endothelial cells which contain large pores without any diaphragm. These endothelial cells allow rapid flow of plasma but prevent the passage of formed elements of the blood, that is the cellular components, the white blood cells, red blood cells, and the platelets. And what is the role of the JBM? Now, a more elaborative, more descriptive role of the GBM here. This common basal lamina stops the passage of the macromolecules. How? In three ways. According to the molecular size. The molecular size is more than 10 nanometer, that molecule cannot pass through the GBM. And according to the molecular weight, if the weight is greater than 69 kilodaltons, these molecules are restricted. So albumin restricted, but smaller molecules like simple sugar, metabolite, amino acids, and even small peptide can pass through the GBM. Presence of negatively charged glycosaminoglycan heparin sulfate in the GBM restrict the negatively charged molecules, and unlike albumin, the cation like sodium ion can pass through the GBM. The role of the slit diaphragm. The slit diaphragm are porous but allow the passage of very small molecules only. Macromolecules like albumin and gamma globulin cannot pass through the slit diaphragm.